My name is Timber Wolf, and I'll be teaching you about DNA replication, transcription, and translation in prokaryotes. In prokaryotes, DNA can be replicated in two different ways. The first way is beta replication. Beta replication is a bidirectional synthesis of new DNA. So we will begin with a double-stranded circular DNA. At the replication origin, the double DNA strand will begin to unwind. It will then produce a replication bubble, and at either end of the replication bubble, there is a replication fork. The replication fork will move around the circle and eventually produce two circular DNA molecules. Rolling circle replication is another way prokaryotes can synthesize new DNA. First, you begin with the double-stranded circular DNA. The circular DNA will then cleave off one of its single strands. The single strand will then begin to replicate from the 5' end to the 3' end, and will work its way around the circle. Once it has worked its way all the way around the circle, you will then have to cleave off the newly replicated DNA. The DNA will then be a linear DNA, and then it must reform back into a circular single-stranded DNA. This single-stranded circular DNA will then be used as a template to form double-stranded. In prokaryote, transcription begins with the promoter region. The promoter region contains two consensus sequences. They are known as the Tata and the Tata-Gaka. In between these sequences, the sigma factor, which is this, will then bind between the sequences, and then the RNA polymerase will then bind afterwards. So the RNA polymerase will then move down the DNA with the sigma factor transcripting the newly synthesized RNA. The RNA will then be synthesized from the 5' to the 3' end. Once it reaches the terminator sequence, the sigma factor will release from the complex. Following the sigma factor, the RNA polymerase and the newly synthesized RNA will unbind. In prokaryotic cells, translation occurs in the nucleoli. In eukaryotic cells, translation occurs in the cytoplasm. So for translation to occur, you first need to begin with the 30S ribosome. This 30S ribosome will have the help of initiation factor 3 to bind it to the mRNA. This initiation factor 3 will bind the mRNA in a way that the start codon AUG will line up in the P site. So this mRNA will have a consensus sequence known as the Shin-Dallarton, which is upstream from the start codon. So once this complex is formed, a tRNA with the anticodon to the start codon will be escorted using the initiation factors 1 and 2. So this codon will translate to formyl methylamine, and in eukaryotic cells, it will only translate to methylamine. So once the tRNA is bound to the start codon, the 50S ribosome will then come in and attach the 30S and the mRNA complex. Once it has bound to the complex, all initiation factors will be removed from the complex. Cellular elongation starts when a tRNA with the anticodon sequence to the codon that is located in the A site binds to that codon. So once it binds to that codon, you will then have peptidyltransferase, which is an enzyme, and it will cut the amino acid from the tRNA that is located in the P site. It will then form a peptide bond between the two amino acids and adding it to the amino acid on the A site. Then you will have G-translocase, which is an enzyme, and it will shift the mRNA by each codon. So the codon that is located in the A site will move to the P site, and the codon located in the P site will move to the E site. Once the mRNA has been fully translated, it will reach a stop codon that it will be recognized by a releasing factor. This releasing factor will then release the newly synthesized protein. So once it releases the protein, initiation factor 3 will come in and bind to this complex of the 50S, 30S, and mRNA. So when it binds, it will unbind the 30S, the 50S, and the mRNA so that they can be used once again for translation.